हाँ चलो. Good morning, students. Today we'll take up lesson number 12, that is biotechnology. The weightage of this lesson is 5 marks. Compulsory you will have to attend, and 7 marks is with option. Now let's understand what's biotechnology. We know the word bio, it means life. Okay, and technology is a technology that uses biological systems or living organisms to create or modify products for a particular purpose. That is biotechnology. Okay, again I will repeat, what is biotechnology? Bio means life. So the technology or the techniques that is utilized, that is utilized or used for the purpose of any modification. Okay, that is known as the biotechnology. It is a technology that is derived from living things and their natural processes, right? Now, the general categories in which field, various fields where this biotechnology is used are like medical, industrial, environmental, and agriculture. These are the 4 main branches. In the later slides, I will be telling you about, in the next slides, I'll be telling you about that in detail. Okay. So the general categories are medical biotechnology, which includes vaccines, diagnostics, that is how the diagnosis process, then the pharmaceuticals. Sorry. Industrial biotechnology, enzymes and microorganisms for processing the products. Then environmental biotechnology for microorganisms for bioremediation. Okay. Then agriculture biotechnology, that is to enhance the crops, feed, and the fertilizer. Crops, feed, and fertilizers. This biotechnology is the branch of applied science that utilizes organisms and their derivatives in order to produce products and processes. A very important definition which includes all the things, students. Okay, I repeat, biotechnology is the branch of applied science that utilizes organisms and their derivatives in order to produce products and processes. These products and processes can apply to several aspects of economy, ranging from healthcare and medicine to biofuels and environmental safety. Let's see how. Now, biotechnology is defined as the manipulation of biological organisms to make products benefiting human beings. Okay. So whatever changes, whatever techniques are being adopted or being discovered or invented, that is all for the well-being or for the benefits of the human welfare. Okay. With the discovery of the structure of DNA by Watson and Crick in the 50s, biotechnology has extended into the areas such as gene transfer through direct manipulation, then human stem cell research, cloning, fusing of different types of cells to produce medical products. Okay, so DNA structure you're familiar with. I have told you how replication of DNA takes place. This DNA structure, that is the double helix structure, it was first put forth by Watson and Crick. So on that basis, these further developments like the biotechnology has extended into various such fields. Then biotechnology applies the knowledge of biology to enhance and improve the environment, health, and food supply. Then using biotechnology, scientists work to develop environment-friendly alternatives to fossil fuels and plastics, new medicines, vaccines, and disease diagnostic tools, higher yielding and more nutrient-rich crop plants. So these are all possible because of this new techniques, that is by using biotechnology. Okay, depending on the tools and applications. Tools means what? Some requirements are there, like that, like enzymes are there. Okay, then nuclear restriction enzymes are, certain enzymes are there. Then the one which donates and the one which is the carrier or which works as a vector, like an agent. So these all come under the tools. Okay. So depending on the tools and application, if it often overlaps with the related fields of bioengineering, biomedical engineering, molecular engineering, etc. Then, traditional biotechnology. Now, there are two biotechnologies you'll be studying. Very clear difference is there. One is traditional and one will be the medical. Okay, a modern technology. Clear? So it refers, that is, a traditional one refers to the using living organisms to yield new products or modified foods or other useful products for human use, right? Which includes the breeding animals and crops to make cheese or yogurt, bread, beer, and wine. Techniques like selective breeding, hybridization, and mutagenesis are used in current applications of biotechnology. These all come under traditional student. This is a routine, traditional, this is a routine type of biotechnology methods. Okay. Then other technologies include fermentation, food processing, tissue culture, etc. The diversity of microorganisms and development of genetics expanded the potential of traditional biotechnology. So because of all this thing, development in the field of genetics, because of the diversity of the microorganisms, etc., etc., from traditional biotechnology, it was switched on to the modern technology. Okay, there is some improvement to that. Clear? Right. This technology is based on active techniques which have great efficiency and accuracy and are cheaper. Modern biotechnology. It involves the use of GE techniques. What is this GE? It is genetic engineering techniques, okay? Such as recombinant DNA. I have already told you when I was teaching about the DNA, etc. Okay, functional and structural genomics, then DNA diagnostic probes also, and other methods for genetic modification. All these terms I have been used before in earlier lessons which I have taught you. So you should be familiar with this. You should try to recall, students. Okay. Currently, the techniques are being used in many areas like food, agriculture, forestry, healthcare, environment, minerals, and industrial processes to develop the new products and the processes. Okay, students, see, there was a traditional method. Okay, there was a traditional method, but there has been some improved. Something has been improved from this traditional to it has switched on to the modern technology. Okay. Then one of the most extensive application of modern biotechnology is in agriculture. Recombinant DNA techniques and mutagenesis are used to develop plants with novel traits. Biotech diagnostics have been used to detect a wide variety of diseases and genetic conditions. Now, see, understand this. This modern technology is not only to improve the variety, not utilized only to improve the variety in various fields, okay? But also it is able to recognize the diseases and the genetic conditions. It is able to recognize or find out the genetic conditions of that particular organism, whether a plant or an animal, okay? Now, see, this is the difference between the traditional, or we can say comparison chart of traditional biotech, that is biotechnology, and modern biotechnology. Now, let's understand what is traditional biotechnology. We will compare these two things, okay? Now, traditional refers to the traditional techniques of using living organisms to yield new products or modified foods or other useful products for human use. Okay, this is traditional one. I will tell you, like curdling of the milk, correct? Then here in modern, the diversity of microorganisms and development of genetics expanded the potential of traditional biotech. It expanded the potential of the traditional biotech, and that becomes the modern biotechnology. Okay, and ultimately led to the development of modern biotechnology. Then examples of early biotechnology includes recombinant DNA techniques, tissue culture, and mutagenesis. Then examples of modern biotechnology include DNA profiling. I've told you what is DNA profiling, fingerprinting, that is, okay, genome analysis, then transgenesis, then DNA cloning, tissue engineering, etc. Third point is breeding animals and crops to make cheese, yogurt, bread, beer, and wine. Microorganisms are used to produce various products such as enzymes for use in laundry detergents. Here in modern biotechnology, techniques are used in many areas such as food, agriculture, forestry, healthcare, environment, minerals, and industrial processes. It involves use of natural organisms to create or modify food or other useful products for human use, whereas in modern biotechnology, it involves manipulation of the genes. I hope you will understand the difference very clearly over here. Here they're using the organisms in a natural way. Okay, whereas in case of modern biotechnology, it involves manipulation of the genes and living tissues in a controlled environment. You will see certain experiments for the process, how they have been, how the modern biotechnology is utilized. Here, there are two main principles of biotechnology. One is the genetic engineering, another is the chemical engineering. In genetic engineering, the technique, it is the altering the chemistry of DNA and RNA so that it can be introduced into the host organism to change the phenotype of the host organism. Okay, so when there is some changes made in this DNA and RNA, the chemistry or the molecular structure of this DNA and RNA, and then it is introduced into the host organism, and which will change the phenotypic appearance of that host organism. This is genetic engineering. Okay, changing the genetic material. Fine. Now chemical engineering. In this, it deals with the maintaining sterile conditions to enable growth of only the desired microbe or eukaryotic cell in large quantities for the manufacture of antibiotics, vaccines, enzymes, etc. Okay. Now you see, in genetic engineering, what is done? The genetic material itself is altered, is changed. Okay, which brings about the complete change in that particular host organism. Yeah. Whereas in chemical engineering, what is done? It will only the desired character, only the desired part is made used of it. Then, genetic engineering is biotechnology involved in synthesis of artificial genes. I told you, change in the genetic material. Okay, so artificial genes, repair of genes, deletion of the genes, and shifting of the genes. Because of this, using artificial repairing of the genes, then deletion that which is not required, and shifting of the genes which is required in a particular part. So these things which is possible only in genetic engineering. Okay, production of recombinant DNA and manipulating them for improvement in human beings, plants, animals, and microbes. So genetic engineering, broadly, what does it mean? It means that there is a manipulation, okay, in the genetic material according to the need or according to the type of improvement that is required in case of human beings, maybe plant, in case of human beings, in case of plants, animals, and microbes. Okay, so this is genetic engineering. Then an important aspect of genetic engineering is recombinant DNA technology, which is nothing but the gene cloning, in which the desired DNA sequence from different sources are obtained, and then they are joined together to form a new combination, and this is known as recombinant DNA. Understand this properly, student, that whichever characteristics we need, okay, so that segment of DNA is selected because genes are present on that, right? So that particular segment is selected from various sources, or we can call it as from different sources. Then they are joined together, okay, and then when they are joined, collecting from different sources, obviously a new combination will be formed. This new combination which is formed is known as the recombinant DNA, okay, which gives the product which we are actually aiming for or working for, that is the desired product, right? It is also called gene manipulation or gene splicing. Splicing means they are cut, they are separated. Okay, with the help, how they are cut? With the help of endonuclease enzyme. This endonuclease is also considered, is also known as the scissors of the genetics. Okay, it is the scissors of the genetics. We call it as a restricted endonuclease also. Why? Because endonuclease, I told you ASE, it indicates for the enzyme. Why restricted? Because how much we require, that much can be cut because of this particular enzyme. And then a combination of various desired genes is made, and this particular DNA that is known as recombinant DNA. Okay. Next, polymerase chain reaction. It is abbreviated as PCR. There is a machine, student, that it is known as a PCR machine. Okay, it is a technique that results in exponential amplification of a selected regions of a DNA molecule. What we have been talking about just now, that the desired characters which are present in that particular part of the DNA segment, it is cut and then they are joined, forming a new DNA molecule, okay, which is required of the desired product. Clear? Now here, how they are cut, how it is multiplied with, it is amplification. Exponential amplification means what? The fast, quick multiplication of that. Okay, for this we require, there is a machine known as PCR machine. The full form of PCR is polymerase chain reaction, and the idea of this PCR is credited, or it is by Sir Carey Mullis. Okay, he's a research scientist at California Biotechnology or Biotech company, Citrus, in 1983. Okay, the PCR is one of the most important inventions of the 20th century in molecular biology. Yeah, it is a technique that results in exponential amplification of a selected region of a DNA molecule. So the selected part of the DNA molecule, that is very fast, very quickly, it can be multiplied. Quickly multiplied is the meaning of exponential amplification. Now here you see, this one you can see here down, this is the thermocycler, or we can tell you the PCR machine it is. Okay, and these are the components, these are the required things in this machine. Clear? And each cycle takes about 3 to 4 minutes, okay, to complete the process. Now we will understand with this first. Okay, how does it process? This is one particular cycle, and each cycle takes approximately 3 to 4 minutes. Okay, now here you see, this is the DNA strand. Okay, in 3 steps it will take place. One is denaturing, other is annealing, and other is extension. Now here during this process, student, that is PCR process, that is polymerase chain reaction process, what happens? Alternately, the 3 steps is there. Once it is very hot, okay, that is 95 degrees centigrade, then it becomes cool. It is allowed to cool at 55 degrees centigrade. That is, the temperature is brought down, and again to synthesize the new strand, again the temperature is high, that is 72 degrees centigrade. Okay, now here you see, this is a DNA strand. Okay, it is separated, they are separated. So you can see this dark color and light color. Okay, this is one strand, this is another strand. Now you see, under in the presence of 95 degrees centigrade, what happens? These two strands, they separate here. And this process is known as denaturing. That means it is separated. Its original position is changed. That is denaturing. Okay, and now you see, once it is denatured over here, or we can say the strands have separated, what happens? Then the temperature is brought down to 55 degrees centigrade. Okay, and a primer is attached to that, to either side. See, this is a primer. Okay, primer is attached, and now this extend by adding the DNA polymerase. Okay, that is different types of purines and pyrimidines. You see here this nucleotide. I told you all these things are required for the process of PCR processing. Okay, now here this is added one by one. Again, when it is adding, the temperature required is high, that is 72 degrees centigrade. Then only it will synthesize the new strand. So it will extend. Two sides are required. One is forward, okay, one is forward side, another one is the this thing reverse side. Okay, so two primers are required for each strand, one for reverse and one for the direct, that is a forward. Clear? Now you see, and then they polynucleotides, they are attached over here, and then that means the triphosphates, they are attached, and then you see a new strand is formed over here. Okay, now you can see this, and this is known as the extension. That is, see, now one new strand is formed over here. Now let's read this point. What is the basic requirements of PCR technique? DNA fragment to be amplified. See this. This is the DNA material or DNA fragment which is to be multiplied or which is to be amplified. Okay. Then we need two primers. We need, see, these are the two primers. Okay. One which is moving in a forward direction, another which is moving in a reverse direction. Here. Then a thermostable DNA polymerase enzyme that can withstand temperature up to 94 degrees centigrade, and which can, that is the Taq polymerase. Okay, what is this Taq? Taq means Thermus aquaticus. It is a bacteria. Okay, this is acquired from the bacterium known as Thermus aquaticus, right? So Thermus T is taken and AQ is taken of aquaticus. So here they say it as a Taq polymerase. Please understand this. Okay, Taq polymerase, isolated from bacterium Thermus aquaticus. Clear? So a thermostable DNA polymerase is required, which can withstand the temperature 94 degrees centigrade. Then 4 types of DNTPs are required. See here, 4 types: cytosine, guanine, adenosine, and thymine. These nucleotides, they are required. Okay, so the chain will increase. The chain, it will keep on adding. Okay, and then a new strand will be formed. Clear? What is the full form of DNTPs? It is deoxyribonucleoside. Deoxyribonucleoside. T means what? Tri, and P indicates what? Phosphatase. Okay, DNTPs, deoxyribonucleoside triphosphatase. You should know the full form of it. When you use the abbreviation, at least once you should mention the full form of it in your answer. Okay. Now let's understand this. What is the principle of PCR? These are the basic requirements. See, these are the basic requirements or the components for PCR. This is the process I told you. It takes place in 3 steps. Okay. Now what is the principle of PCR? This should be known first, but I thought first you will understand the process. So this will become more easy for you to understand. Okay. Now the double-stranded DNA of interest is denatured to separate into 2 individual strands. I told you this process. Then each strand is then allowed to hybridize with a primer. That is renaturation. Now it is separated. Now again it will for uniting that, for reuniting. So renaturation, that is the annealing. Okay, see, the primers are used. Then the primer template duplex is used for DNA synthesis. The enzyme is the DNA polymerase. Okay, these 3 steps, that is denaturation, renaturation or annealing, and synthesis, are repeated again and again to generate multiple copies of target DNA. So this is the principle of the polymerase chain reaction. What are the basic requirements are here, and this is the procedure. Now once again, this is the PCR machine. These are the components of the PCR. Okay. Now see the next slide, again the same thing is repeated, but what you will write, for that I have given you this slide over here. Okay, mechanism of PCR. The PCR methods use thermal cycling. What is the thermal cycling? I told you, once it will be very hot, once it will cool down, and again it will be, the temperature should be high. So alternate heating and cooling DNA sample should be there. Okay, each cycle has 3 steps. Heat denaturation. Why heat denaturation, student? Because of the temperature, the strands will be separated. Okay, here the temperature required is 92 degrees centigrade to 94 degrees centigrade. This breaks the hydrogen bonds. When this much temperature is there, then only the hydrogen bonds which join the 2 strands of DNA will be separated. Okay, sample to get a single strand DNA, right? So you may get question in 1 mark or 1 sentence type that why so much temperature is required, then you will write this particular statement. Yeah. Then annealing or renaturation. Okay, that means sequence specific primers are linked to the DNA segment by lowering the temperature to 55. Now, because of this high temperature, they have separated. So if we keep the same temperature, will it join? No, because that much temperature is required to separate, not for joining. So it has to cool down. The temperature has to be brought down. That is approximately 55 degrees centigrade. And then again, the linking part will start. Then only the linking of the DNA segment will be there. Okay. Then extension. Now it has linked with the help of the primers. Okay. Once direct and once in forward direction, another one in the reverse direction on the 2 different strands. Now this has to be extended. Now to extend the temperature, it is raised up to 72 degrees centigrade to 75 degrees centigrade. Okay. Of the DNA strand is done by addition of DNTPs. I told you what is that, DNA polymerase, that is, okay, deoxynucleoside triphosphatase. Behind the primers, they are added, and as the primer moves forward, these phosphatase they are added to that. These nucleotides are added to it, okay, with the help of DNA polymerase enzyme. And this takes place at the temperature 72 degrees centigrade to 75 degrees centigrade. And each cycle will take about 3 to 4 minutes approximately. I hope you have understood this very nicely. 3 to 4 times I have told you. Now, what is the application of PCR techniques? Okay, we have been studying about the PCR process, how it works, how is the mechanism. Now, what is the application of this technique? It is used in DNA cloning, gene amplification, cloning again, the formation of many DNA molecules. Okay, then gene amplification, that is multiple forming, getting more and more quantity or number of genes. Then diagnosis of hereditary diseases. Then mutation detection, identification of genetic fingerprinting. In forensic science, it is used. Scientific tests are done. Techniques used in connection with the detection of the crime. So that is how it is used in case of to study the forensic in field of forensic sciences. So these are the various applications where it is used. These techniques, they are used. Then I told you about the tools. There are tools like enzymes, vectors, target DNA, or foreign DNA. Now, enzymes, various enzymes are used, that is restriction endonuclease, like DNA ligase, reverse transcriptase, DNA polymerase, alkaline phosphatase, etc. These are the various enzymes which are used. Okay, then vectors. A vector is a carrier of DNA molecule to which the fragment of desired DNA is attached, and they may be plasmid DNA. Then bacteriophages, I told you, the bacteria, that is E. coli bacteria, is used as an experimental bacteria, okay, or the organism. DNA of plant and animal viruses. Then shuttle vectors. What is this shuttle vectors? Vectors with 2 origin sites. I told you when DNA molecule, when the DNA, it separates, the 2 strands separate, it separates at a particular part, okay, and that part is known as the origin. So like this, if 2 origin sites are the origin site, that is which can propagate 2 different host species together. And what does ori means? It means the origin of the application. Okay, and then target DNA, that is, or we can call it as a foreign DNA, desired fragment of DNA, that means which we want, okay, which is passed from one organism into another by different vectors, is called the target DNA. Then, what is the method for this rDNA technology? These are the various steps, students. Okay, isolation, the one which we want. Okay, isolation of DNA, that is the gene. DNA consists of genes. So whichever part we need, that much, whichever gene we need, we cut that. Okay, from the donor organism. Then after taking it from the donor, it has to be inserted. Okay, and then it has to be transferred. Then it has to be set down. It will be of the donor also. Please wait. Let's continue. So these are the various steps, students, which are required. We'll see in the next slide in detail. Yes. Now here you see, this is how it is done. This is the donor cell, okay? Its DNA molecule is extracted. It is the desired genes, they are cut into pieces this way, right? This is one part. The other one, a vector is required, someone which has to carry all this thing to the experimental host, okay? Now here you see vector, this vector can be a plasmid or it can be a bacteriophage, right? Now this is the vector, it comes over. With the help of the restriction enzyme, okay, that is endonuclease, this is cut, okay, the DNA of this vector is cut. If, insertion, it has to be inserted. Now this part, that is a desired gene, is inserted here, right? Now this vector DNA, which is having the target DNA, now this is coming over here and it has to be inserted into the host bacteria. And I told you, usually in host bacteria, we use the E. coli bacteria. E indicates for Escherichia coli. The name of the bacteria is Escherichia coli bacteria. Even when we study about the prokaryotic cell, we use Escherichia coli. And I think you're familiar with this. If you are listening to the lectures or reading the books properly, you are familiar with this Escherichia coli. It is abbreviated as E. coli. Okay. Now this introduction is done. Okay. It is done in this host cell. Now see, it comes over here. Now see here, this is the transformed one, okay, the one which is cloned over here. Now it has been which has been the target DNA. Now this will allow to multiply. See, identification and selection, amplification, it will be done. Now see, student, this will be multiplying, but along with this, even the host cell, the DNA will be multiplying, isn't it? So that I will tell you when I tell you the steps again. Now this is the diagrammatic representation. If it is asked, you will have to make this, right, and have to write every step of it. Then here cell multiplication is taking place. Then protein production from cloned genes have taken place. Now this will be harvested. The one which we require, the protein which we require, will be harvested. Okay, so this is how it is. The process is of our DNA technology. Now in detail here you see isolation. I told you those steps, isn't it? So each step, if it comes in writing, if it comes, this question comes, what you're going to write in that. Isolation of DNA from the donor organism. The desired DNA, which was nucleotide sequence of choice, is cleaved. Cleaved means it is cut from the donor by using restriction endonuclease enzyme. Okay, now it is inserted. The separated DNA is added or inserted to vector, vehicle DNA, which is usually, it is also known as vehicle DNA because it is carrying from there, okay, which is usually plasmid DNA or bacteriophage DNA, right? The vectors do not disturb the nucleotide sequence and carry this foreign DNA. That means what? This agent or this vector or this vehicle, which is carrying this DNA, it will not disturb the nucleotide sequence. It will only work to transfer or to carry that particular chosen DNA to the host cell, okay, for amplification or for multiplication. Understood? But it will not disturb the sequence which was inserted here. Using enzyme DNA ligase, the foreign DNA and plasmids are united together. This process is known as ligation. Now remember this, the enzyme which is used to cut the DNA molecule is the endonuclease. See here, endonuclease. And the enzyme which reunites or which helps in uniting the DNA is the DNA ligase. And the process is known as ligation. And the intermixing of different DNA strands is called recombination. That means different DNA strands, they have been recombined. Okay, so the process is known as recombination. Then this, that is the DNA. Okay, this plasmid is now called recombinant plasmid. Vector DNA with new introduced gene is called rDNA, that is recombinant DNA or ceramic DNA. Please read this again and again. I'm trying my best to explain you. Okay. Now next step, now it has been inserted. Now what will be the next step? That it has to be transferred into the host cell for further multiplication. Okay, but you see, all the bacterial cells, they will not accept this foreign DNA easily. So for that, these bacterial cells, they are treated. Okay, they are the process used for the treatment is thermal shock, calcium ion treatment, or electroporation, etc. Okay, so these bacteria, they may not accept it immediately, but so they are given some treatment so that they accept this foreign DNA easily. Okay, and this plasmid is inserted into the organism. Okay, now it is inserted into the bacterial cell, that is E. coli, and this process is known as transformation, which has the capacity to multiply rapidly. Usually, the organism is a bacterium E. coli, which should be non-pathogenic. Okay, that means it should not have any type of problems or difficulties or diseases, etc., or any type of pathogens in it. It should be harmless and is easy to cultivate. Okay, bacterial cell with our DNA is called transformed host. Understood? See this slide, I will tell you. This one, now here you see the vector DNA is added over here. See there? Now this is known as the transformed host. Okay, this is at this stage it is known as host bacteria. But when this is introduced, then it is known as transformed host. Right? So transfer has taken place. Now what happens, student? Even the bacterial cells, the original DNA, that is also multiplying, and this is also multiplying, so it gets mixed up, isn't it? So because it gets mixed up, what we need the desired ones, which we have inserted in that, isn't it? So for that, now selection is to be done. So that is known as selection of the transformed host cell. How the procedure is, see, the selection process involves filtering and transformed host cells only. Okay, as the transformation process generates a mixed population of transformed and non-transformed host cells, so we need to filter that. Clear? For this process of isolation, a marker gene of plasmid vector is employed. The marker gene, like PBR322 plasmid vector, contains different marker genes. Okay. Ampicillin resistant gene is there and tetracycline gene is there. So when PHT1RE, this is that vector, marker gene, okay, it is used. It knocks out the ampicillin. So when this is used, it will knock out the ampicillin resistant genes, now which are able to resist, okay, from the plasmid, so that the recombinant cell becomes sensitive to ampicillin, and this way they separate the desired genes. Okay, selection of the transformed host cell, this was. Then multiplication, now that has been transformed host cell, that DNA that is separated, now they have to be multiplied. The transformed host cells are introduced into the fresh culture media now, and the host cell divides and redivides along with the replication of the recombinant DNA they carry. Okay, then expression of the gene to obtain the desired product. The desired product is separated and purified through downstream processing using suitable bioreactor. So these are the different steps which are required to form a recombinant DNA or to get the desired quality, desired type of DNA. Now, what are the applications of biotechnology? Now that part is over. Here you see the biotechnological application. In which are the different areas? It is in the field of agriculture, medicine, transgenic animals, ethical issues. Ethical issues means what? Suppose if this biotechnology is used, will it harm the nature? Will it harm the animals? Will it harm the living creatures, living beings, etc.? So that all comes under ethical issues. Okay. Then what is this transgenic animals? Transgenic animals means what? The animals which has been inserted, in which the new genes they have been introduced and they are transformed. Okay, so that is transgenic animals, or it can be transgenic plants also. In the field of agriculture, in the field of medicine, in the field of biosafety issues. Now, Here you see biotechnology in 4 different colors. It has been mentioned over here. When it comes with the red color, it indicates for the medical biotechnology is biotechnology which is applied to manufacture pharmaceuticals like enzymes, antibiotics, and vaccines, and is used for molecular diagnostic. So see how biotechnology is used in case of medical field. Okay, and this is referred as the red biotechnology. Then when it comes to the white biotechnology, or it is otherwise also known as industrial biotechnology, is biotechnology which is applied to industrial and other production processes. Okay, like beer, this liquors, beverages, acids, etc. Yeah. Then green biotechnology, when we use the word green, understood that it is concerned with the agriculture biotechnology. It is biotechnology applied to agriculture processes and products. Okay. Then blue technology, again, marine biotechnology, the color itself indicates that it will be water. Marine biotechnology, it is marine and aquatic applications of biotechnology. So these are the 4 major branches that they have mentioned with the help of the colors. Okay, red means medical, green means agriculture, white means industrial, and blue means the water, that is the aquatic or the marine. Then, now you see, the knowledge, the roots are here. Okay, the roots are here, which all together form what is known as biotechnology. But because of by the use of this knowledge over here, okay, by the use of this knowledge over here, this biotechnology has developed many more branches. Understood what I'm telling? Because of the root knowledge over here, the biotechnology has raised, because of this knowledge, the biotechnology has given out many more branches have been developed. Okay, these branches can be recombinant DNA technology, environmental technology, waste utilization technology, then food and feed technology, then biomolecular engineering, then plant and animal cell culture technology, biomining, and then fermentation technology. So the knowledge is there. This all comes under biotechnology. Because of this knowledge, the more and more branches they have been flourished or they have upcome. Next, healthcare biotechnology. It refers to a medical or diagnostic product or a vaccine that consists of or has been produced in living organisms and may be manufactured via recombinant technology. Then medical biotechnology is the use of living cells. See, medical in case of medical science, that is medicines or pharmaceutical. Bio means life. Technology, the technique. Okay. So it will be by the use of living cells and living materials to research and produce pharmaceutical and diagnostic products that help treat and prevent human diseases. Human insulin is one. It is a protein hormone that is used as a medication to treat high blood glucose. Now, human insulin is a bioengineered insulin, very similar to insulin made by the human body. In human body, it is produced by beta cells of islets of Langerhans of pancreas. The first genetically engineered synthetic human insulin was produced in 1978 using E. coli bacteria. Insulin producing gene from beta cells were isolated and introduced into E. coli. Understood, student? Insulin producing gene from the beta cells present in the pancreas, okay, they were isolated. That means they were separated, and where they were introduced into the experimental bacteria, that is the E. coli. Now, in E. coli, what happened? The gene undergoes transcription and translation to produce insulin, right? This insulin is isolated from E. coli. It was packed and was used for human welfare. Understood? By the knowledge of biotech, we have introduced the gene into E. coli and transformed this E. coli into a factory which provides us or synthesize insulin. See how biotechnology is in you. Very interesting subject it is, students. Read properly. If you don't understand, ask me through WhatsApp. Okay. Then vaccine, how vaccine is produced. A vaccine is a biological preparation that provides active immunity to a particular infectious disease. We all know that. Okay, a vaccine typically contains an agent that resembles a disease-causing microorganism and is often made from weakened or killed forms of microbes. Okay, its toxins or one of its surface proteins. Now, see, we all have taken vaccine against the coronavirus, isn't it? And how it is see how it is being produced. Two vaccines they are produced or they are manufactured in India itself, and we are supplying to other countries also. So I told you after reading the first sentence that you understand this. A vaccine is a biological preparation that provides active immunity to a particular infectious disease. Okay. Oral vaccines are that, student. Oral vaccines is what, like polio vaccine, which is given to babies. Okay. Not necessary that all vaccines should be injected only. So it can, instead of injecting, it can be given orally also, that is through the mouth also it can be given. Best example is the polio vaccine. Okay. Then next, that is we take vaccine means we take the protein. Where there is a protein means there should be a gene which is transcribed and translated. Proteins were isolated from the pathogen or virus and was introduced into the plant. Okay. In plant, this gene undergoes transcription and translation, and the plant produces the protein, that is the antigen, and the edible vaccine was developed. Example, banana, very good example it is given, that is of banana. What happened, students, in banana that this gene was introduced. Okay, and when banana was eaten, the antigen entered the body automatically and antibodies are developed. Okay, so when the plant is growing itself, before that itself, when these genes are introduced, it comes up, it transcribes and it translates and it multiplies in, or it is present in the fruit. When we eat that fruit, then automatically that is entering into the protein is entering into our body. Okay, which help us protect from any type of disease or problems. Clear? Therefore, we can say that edible vaccine is a genetically manipulated food containing organisms or related antigens that may provide active immunity against the infection. But simply just I want to tell you here, that is that not necessary that we should take through injection only. The technology has been so great that we can take it through the fruits or the food that we eat. Okay. An exciting invention is production of melt in the mouth vaccines that can be administered by placing it under the tongue. So this can be utilized by using that particular vaccine in the form of, maybe in the form of a tablet. Okay, it is kept under the tongue, and then slowly it can be it melts up in our mouth, and which will flow throughout, which will go towards to the different parts of our body through the bloodstream. Okay, and this type of vaccine, that is the flu vaccine, it is formed by the flu vaccine by Bacillus thuria. Right? Now here, medical biotechnology, you can see so many areas are open with that. DNA fingerprinting, you know this, that is DNA profiling, then gene profiling, prenatal diagnosis of inherited diseases, then pharmacogenomics, then skin grafting, development of the vaccine, then biopharmaceuticals like insulin, somatostatin, then interference, these are the hormones. Okay, all these things can be developed, right? Gene therapy is done in which gene is used. Okay, gene is replaced, the harmful gene or the disease gene can be replaced in an organism, okay, by a good and a healthy gene, that is known as the gene therapy. Clear? So these are the various fields where medical biotechnology is there. Then biotechnology in agriculture. Now, we studied about the medicine. Now in agriculture, first of all, let's see what is the application of biotechnology. There are many applications of biotechnology in food and agriculture, and this we call it as green biotechnology. Okay, like conventional plant breeding is there, then tissue culture and micropropagation is possible, then molecular breeding or marker assisted selection is there. I told you about that marker gene, right? Genetic engineering and GM crops. GM is genetically modified crops. Okay, plant disease diagnostics are there. Then microbial fermentation is there. So these are the applications of biotechnology in case of green biotechnology or in field of food and agriculture. Then what are the benefits of agriculture biotechnology? It is, we see that improved yield from crops we see, then reduced vulnerabilities there of crops to environmental stresses, okay? Then increased nutritional qualities of food crops are seen, then improved taste is noticed, texture or appearance of food is there, then reduced dependence on fertilizer, pesticides and other agrochemicals because it is already modified what is required. So fertilizers, pesticides and other agrochemicals, they are reduced. We may not depend upon them anymore. Okay, then production of the vaccines is noticed. Then biotechnology in gene therapy. I told you a normal gene can be inserted in place of the abnormal gene or the gene which may cause some disease. Clear? Now this can happen in two ways. One, by germline gene therapy, that means in the reproductive cells, that is transfer of a section of DNA to cells that produce eggs or sperms. Okay, so through germline, that is. And somatic, that means it can be in any cell. It can be transferred to any cell of the body. That is the somatic cells only. Okay, so the gene therapy the gene therapy treats a wide range of diseases like cancer, cystic fibrosis, heart disease, diabetes, hemophilia, and AIDS. Then what is GMOs? That is genetically modified organisms. A genetically modified organism is any organism whose genetic material has been altered or manipulated using genetic engineering techniques. Okay, transgenic organisms in use today includes tomato, corn, rice, sheep, cow, and mice. That's why I use the word transgenic organisms. Okay, which will include both plants and animals. Clear? Then this creates combination of plants, animals, bacteria, and virus genes that do not occur in nature or through traditional crossbreeding methods. So they will not appear naturally, but through genetically modified organisms, we get all these changes and the benefits. Then transgenic plants here. First transgenic plant produced was tobacco. The aim was to introduce a new trait in the plant. The inserted sequence is known as the transgene. That the trans indicates what? Transporting, okay? Then, or transformation. Then, purpose is to make plant useful and productive. What was the advantages of transgenic plant? Increased and improved nutrients, longer shelf life. That means shelf life means what? Where suppose you keep it in a shelf. So how long it may get spoiled also, okay? When it is in a original one, but when it is modified, okay, when it is modified or it is biotechnology is used to insert certain genes which will help it to last longer, they remain fresh for a longer period. Okay, then taste is and quality is noticed, then reduced maturation time is there, so that we can get good economic benefit also will be there. When the maturation time is reduced, before time we can bring it up in the market. Then higher yielding crops are there, more efficient use of lands are there, then improved resistance to disease, then increased food security for growing populations and growth challenges. That means the population is increasing, so the amount of food should also increase to fulfill the need of the growing population. So that is also possible. Then advantages of GM food plant. GM is genetically modified. GM crops have improved resistance to disease, pests, insects, and herbicides. They also have improved tolerance in cold or heat, drought, and salinity. That means less scarcity of water, that is drought, and more of salt, that is salinity. Okay, or less of salt. Then insect resistance is achieved by incorporating into the food plant the gene for toxin production from the bacterium Bacillus thuringiensis. I have already told you this, BT crops. Improved nutritional quality, that is biofortification. You are familiar with this biofortification word also. Then modifications in post-harvest characteristics. Genetically modified tomatoes are called flavor saver tomatoes. This is one of the examples that I'm mentioning over here, were developed through the use of antisense RNA to regulate the expression of the enzyme polygalactoronase. Okay, that is PG, abbreviated as PG, in ripening of the tomato. They remain on the vine until, vine means bale, that is, it is weak stem, so the weak stem plant that is known as the vine. Like we have the grape vine. Okay, see the spelling is VINE and not WINE. It is VINE until mature and be supported in a firm solid state. So the tomato, that is flavor saver. Flavor saver means what? The flavor has been saved. With this modification, the flavor, the taste of the tomatoes are not modified, they are saved. Okay, so that is known as the flavor saver. Okay, and this experiment, this was done with by taking tomatoes. Then plants as factories. Now, why we call it as plants as factories? We know factories which gives us many products. Okay, many outputs are there, many products are there in that, various factories are there. But plants as factories means what? Students, plants also give us many certain things like starch, sugar, lipids, proteins. Okay, certain chemical which can be used as perfumes, certain adhesive compounds also. Okay, certain lubricants also. So we call that plants, they are also factories, plants as factories. The recombinant protein produced in transgenic plants include antibodies, metabolites or catabolites, then proteins and vaccines. These are all, we get it when the plants are having the recombinant DNA. Okay, so we call it as a plants as factories. If you get short note, plants as factories, comment, you will write these 3 points. Okay, or any other point if you write, you know it, you can add to that. Every correct point you will give you marks. Okay, antibodies and vaccines against gastrointestinal tract disease, cholera and malaria are known to be produced in transgenic plants like potato, banana, algae and tobacco. Okay, so here antibodies and vaccines against gastrointestinal tract, that means our digestive system. Then transgenic animals. The term transgenic organisms refers to an organism in which there has been a deliberate or artificial modification of the genome. This sentence is being repeated in different forms. Okay, because you must understand very nicely the meaning of transgenic. In other words, a gene from one organism can be inserted into cells from another organism. These transformed cells can be used to grow new organisms. Gene transfer by microinjection is the predominant method to produce transgenic animals. Then applications of transgenic animals, they are used in medical research, used in the field called toxicology as a source of detection of toxicants, then used in biotechnology as they produce specific proteins, then used in xenografting. What is xenografting? The transplant of an organ or tissue or the cells to an individual of another species, that is known as xenografting. Then in molecular biology, they are used for analyzing regulation of gene expression. In the transgenic animals have a great role in pharmaceutical industry. They are used for drug testing and drug production also. Okay, so these are the applications of transgenic animals. Then some examples of transgenic animals are mice. They are utilized for cancer research. Then farm animals, cattle for food production, cattle for human therapeutic production, then sheep, then pigs, fish, chicken. These are all the transgenic animals. Then the 3 terms we'll be studying over here. One is bioethics, one is biopatent, and one is biopiracy. Okay, so bioethics, it is a branch of applied ethics that studies the philosophical, social, and legal issues arising in medicine and the life sciences. In other words, it is the study of ethical issues emerging from advance in biology and medicine. I told you just a few minutes back that when we are using these new methods, new technologies, biotechnologies, when we are using, So we should also take care that it is not affecting our environment, or it is not affecting the living creatures. Okay, so these are known as the ethical issues. Clear? Bioethics means some standards that can regulate the human activities that might help or harm other living organisms. See, we are using it for our benefit. Human being is using it for human welfare, isn't it? But we should also remember, or we should also take care that it should not harm the other living organisms. This is known as bioethics. Okay, genetic modification of organisms can have unpredictable results when they are introduced in the ecosystem, or on their metabolism, or even on humans. So whenever genetic modifications has been taken place, it should be understood that wherever it is introduced, it should be taken care that it is not disturbing the environment, the atmosphere, that is the ecosystem, and neither with their metabolic activities, nor it should affect the human. It should not even affect the humans also. Next, Indian government has set up organizations like Genetic Engineering Approval Committee. It is abbreviated as GEAC. Okay, this committee makes decisions regarding the viability of GM, that is the genetic modified research, and safety of introducing GM organism for public services. Okay, so remember this GEAC, that is Genetic Engineering Approval Committee. Student, there is a committee, okay, which will decide, which will first take care, study that properly whatever new biotechnologies are used and it is to be introduced in the public for the welfare of humankind or for the public services. This committee will take care of it. Okay, that whether it is good or not, whether it will harm or not. So it will take the decision. So to take the safety measures, okay, it will study properly and it will see how much it is useful, and whether it will harm any creature in the environment or whether it will disturb our environment. So this committee will decide. Okay, and the name of the committee is Genetic Engineering Approval Committee. Okay, whatever genetically engineered products are there, that will be approved by this particular committee. So Genetic Engineering Approval Committee. Okay, GEAC. Fine. The major concerns of bioethics are the introduction of genes from one species into the other violates the integrity of the species. Then biodiversity may be affected due to biotechnological experiment, thus posing a threat to the environment. So these are the major concerns what this GEAC, what this approval committee takes into consideration. Okay, that then the third one is that concept of humanness is diluted due to introduction of human genes to various animals, or vice versa, from animals to human beings, right? Then use of animals in biotechnology causes great suffering to them. Then animals are virtually reduced to the status of factory when they are used for production of pharmaceutical exploited for human benefit. So this Genetic Engineering Approval Committee takes these concerns. These are the criteria, or these are the major concerns of bioethics. Then next term is the biopatent. Now, to have a patency, that means suppose there is a person A, there is one person who have discovered something, who has invented something. Okay, so you should know the difference between discovery and invention. Discovery is what? It is something is already present and it has been traced out now. It has been discovered, okay, which is already there, but it was not been able to found out, but now it has been traced out. That is discovery. And what is invention? Something which is newly formed, which was never present, but it is newly formed. So when a person invents something, it for 6 months, it is given, the government right is given to this person, that individual who invents something new, okay? And then it's for 6 months, if nobody claims that they have already invented this, then that patency is given to that person who has invented this newly. Okay, so that is known as biopatent. Okay, a patent is the right granted by the government to an inventor to prevent others from commercial use of his invention. It is personal property of the inventor. Okay. A patent consists of three parts. One, the grant. That means it is the agreement with the inventor. Then specification, that is subject matter of the invention, like what he or she has invented. So that has to be specified. Then claim, scope of invention to be protected. That means that there should be no claim by any other person that yes, they have already done this before this person. So all these things, it is taken into consideration, and if everything is clear, then that person who has invented something new will be given the patency of it. Okay, that is known as patent. Then patents given for biological entities and their products are called biopatent. Okay, so biopatents are awarded by industrialized countries like USA, Japan, and members of European Union. Most of the industrialized nations, students, see, this I am telling you will be thinking why so much of details and all, but first of all, you should know the name of what is patent, then what is biopatent, because it has been invented by using biological entities. Okay, now tomorrow if you are inventing something, so you should know that how do we get this patency of this particular product or with this particular invention. So for this thing, it should not be learned or understood only for writing in during the exam. It should be inculcated in your brain. Okay, that it should be lifelong. This knowledge should be there into your mind. Tomorrow, if you invent something, you can make use of all these things. Okay, you will have a knowledge, already knowledge you will be having. Clear? Most of the industrialized nations are rich financially but poor in biodiversity. Okay, what does this mean? That the natural resources are available in sorry, in one particular country. Okay, but they're not financially rich to make use of these natural resources. Okay, so that is the meaning of this statement, that most of the industrialized nations are rich financially, but they're poor in their biodiversity. Some have lots of money, but they do not have the natural resources, and vice versa. Some have more of natural resources, but they are poor in financial conditions. Okay. Then developing and underdeveloped nations are rich in biodiversity and traditional knowledge of bioresource. Then a patent is granted for an invention, an improvement in earlier invention, the process of generating a product and a design or concept. So see, if you're going for inventing something, remember these points. Okay, and all the best for you all. Next, let's see. Biopatents are awarded for the following. For what it is awarded? Biological procedure or production processes, then new discovered technique, then cell lines, then genetically modified plant or animals, DNA sequences, various biotechnological procedures, products and their applications, strains of microorganisms. So biopatents are awarded for the following things. Okay. Next, biopiracy. The third one, biopiracy, and by this we end our lesson. Unlawful exploitation of the already awarded biopatent is biopiracy, or biopatenting of bioresources of other nations without proper authorization from the concerned nation is biopiracy. That means what? It should not be without taking the consent of the nation where this biology resource, this bioresources are present, then that is the biopiracy. They have to take prior permission. Okay. The advanced industrialized nations are rich in technology and finance, but poor and developing countries are rich in biodiversity and traditional knowledge. So rich developed countries like America are exploiting poor countries like India. For example, in India, neem is being used as insecticide and as medicine since many years, but one of the American company patented neem. Okay. Other examples, plants like black pepper, mustard, turmeric, basmati rice, etc. are also patented by US, United States, that is Germany and Japan. Okay, so this was all that I wanted to tell you. I wanted to make you understand in the lesson number 12, that is biotechnology. Please go through it, understand properly. Any difficulty, you can contact me on my personal WhatsApp number. I stop here. Thank you so much.